Hello everyone. Today we'll be solving Cambridge International ASN A-Level Biology Paper 4 A-Level Structured Questions, May-June 2021, Variant 4.2. Question number 1A. Figure 1.1 is a diagram of part of a neuron membrane at resting potential. So we can see the molecules B, and then we can see A, which is basically a transport protein, and then we can see C, which is going to attach to an active site, and then we can see D, which are some sort of molecules. Now the question says, with reference to figure 1.1, name A, B, and D. So we can see A can be actually a sodium potassium pump, and these molecules, they can be 3 sodium, and typically 2 potassiums are pumped inside and 3 sodium are taken outside. So A is sodium potassium pump, B is potassium ions, and D is sodium ions. Substance C is required to make structure A function. Name substance C. So substance C should be ATP. Some drugs can affect the functioning of the neuromuscular junctions or cholinergic synapses. Table 1.1 lists 3 drugs describes their action on neuromuscular junction or cholinergic synapses. For example, the drug Curar blocks muscle cells membrane receptors at neuromuscular junction. Nerve gas, this inhibits acetylcholinesterase function in synapses. Alcohol inhibits exocytosis of neurotransmitter in synapses. So you see, if we look at the functioning of this particular drugs, the Curar, which blocks the muscle cell membrane receptor at neuromuscular junction, so it will prevent it from receiving signals. Nerve gas will continuous depolarization will occur because acetylcholinesterase will not function and acetylcholine will not be broken down. And in case of alcohol will inhibit the exocytosis of neurotransmitter from the synapse, so there will be no acetylcholine that will be binding with the receptors. So fewer ligand-gated sodium ion channels will be able to open. Question number 2A. All organisms respire. The ATP produced as a result of respiration is used as the energy currency of the cell. Outline 2 examples of movement in cells that use ATP. So muscle fiber contraction uses ATP. The active transport of sodium, potassium ions uses ATP. Movement of vesicles through cytoplasm uses ATP. Exocytosis of neurotransmitter also uses ATP. If there are any flagella or cilia, those that are beating, they can also be using ATP. So any 2 points from these particular points will be necessary for getting the 2 marks. Part 2. ATP cannot be stored in cells, so it has to be continually resynthesized to meet the demands of an organism. A person with a total quantity of 0.2 moles of ATP needs to hydrolyze 150 moles of ATP per day. Calculate how many times the total quantity of 0.2 moles of ATP has to be resynthesized per hour to meet the demand of 150 moles per day. Show your working and give your answer to the nearest whole number. Total quantity of 0.2 moles of ATP. Alright. So first of all, in 24 hours, 150. So we'll divide it by 24. Alright. And then we will multiply it by 0.2. So the answer will be 31.25. Since they want the answer in whole number, so the answer will be 31. So it has to be resynthesized at least 31 times. Name the stages in which chemiosmosis occur in respiration and in photosynthesis. So in respiration, chemiosmosis occur in oxidative phosphorylation. And in photosynthesis, it occurs in photophosphorylation. Part B. Far seals are mammals that are adapted to live in cold temperature. Far seals have large quantities of a type of fat known as brown adipose tissue. Brown adipose tissue cell contains many mitochondria. These mitochondria contain a transport protein called thermogenin. Figure 2.1 shows the role of thermogenin in a mitochondrion of a brown adipose cell when external temperatures are cold. So we can see intermembrane space, inner membrane of mitochondria, all right, and then we can see electron transport chain. H plus ion is being pumped by the electron transport chain. All right, H plus ion can come back through the ATP synthase. As a result, that produces an ATP. H plus ion can also come back through thermogenin, which can produce direct heat energy, which is a great thing for aquatic mammal. With reference to figure 2.1, describe and explain the effect of thermogenin on ATP synthesis. So basically, thermogenin reduces ATP synthesis, which is, I mean, in one sense, for ATP synthesis, maybe the organism does not need enough ATP. However, the proton diffuses through thermogenin, which reduces concentration gradient of H plus ion in intermembrane space. As a result, fewer protons pass through ATP synthase, and more of them passes through thermogenin to produce heat energy. Part 2. When the external temperature is warm, thermogenin cannot function. When the external temperature becomes cold, thermogenin is able to function as a result of cell signaling. Adrenaline is released. Adrenaline acts on brown adipose cells. A sequence of events is triggered that results in the activation of enzyme lipase. Lipase hydrolyzes triglycerides in the cells into fatty acids. Fatty acids that can enter the mitochondrion. Thermogenin then starts to function. Outline the stages of cell signaling that trigger the functioning of thermogenin. So basically, adrenaline binds to the receptor on the cell surface membrane of brown fat cells, and as a result, it activates the G protein. The G protein then activates the adenylyl cyclase enzyme, which is basically a cyclic protein, which is basically a cyclic AMP. The enzyme then produces a cyclic AMP as a second messenger. The enzyme cascade leads to activation of lipase by phosphorylation. Question number 3. One way to measure global biodiversity is to count the number of species of organism. Table 3.1 shows estimates for 2009 of the number of species in some taxa of animals. The number in brackets are the number that were updated in 2019 from the International Union for Conservation of Nature and Natural Resources IUCN for 3 intensively studied taxa. So we can see arthropods with 1.1 million. All right. And then we can see class arachnids, and then crustaceans, myriapods, insects, and other arthropod classes. We can see animals, all right, all of them, and then chordates, mollusks, other animal phyla. List 3 features shared by animal species. So animal species are mostly multicellular. They are heterotrophic, meaning that they have to eat other organisms for food. Vacuoles are small and temporary. Animals have specialized cells. So example, nervous system, eukaryotic cells, all right, and animals are mobile. Any 3 points will give us the full mark. Part 2. Explain how classification of species into a taxonomic hierarchy assists the work of conservation bodies such as IUCN. So basically, classification helps assessing biodiversity. So conservation decisions can be made. Some species may need captive breeding to increase their number. Taxonomic hierarchy is internationally recognized through the scientific community. Table 3.1 shows that the number of species of amphibians, birds, and mammals has increased between 2009 and 2019. Discuss whether the increase in number means that these classes of chordate are being successfully conserved. Yes, because as the species has increased, biodiversity has also increased. All right. Our answer can also be no, because some species only exist in captivity. Guys, because the question said discuss, that's why both points has to be written. You have to also write the point for yes and support the answer, and no and support that no. With reference to table 3.1, identify the class of animals that is most diverse, then calculate the percentage of animal species that belong to this class. Show your working and give your answer to two significant figures. So basically the class of organism, here we can see the best is I would say definitely, there is almost 1,000,000 insect, as you can see over here. Any other organism does not have such a high number. So we're going to talk about insects, and insects have a number of 1024945, and the total organism is 1191770. So into 100% equals to 71%. So 71% animals belongs to insects. Part B, analyzing in Part B, an analysis in 2019. Part B, an analysis in 2019 concluded that twice as many insect species have population that are decreasing in size compared with chordate species. These analyses focused on developed countries that have large human population. Outline two factors that may cause population of insects to decrease in size in developed countries that have large human population. So there can be habitat loss due to deforestation to provide homes for so many people. There can be, it can be due to food shortages. All right, insecticide is used in farming, so the insect did not have an amount of, they died. And also it can be increased due to increased predators or diseases. So guys, any 2 points from this particular answer will give you the full mark. Explain how the introduction of crops that are genetically modified to express the Bt toxin can benefit biodiversity. Bt toxins can only kill specific insect that feeds on the crop, and this reduces the use of insecticide significantly. Other good insects can then survive and help to pollinate the plants and also increase their biodiversity. 4a, genetic technology involving the creation of recombinant DNA can be used to treat different human diseases. These include diseases such as diabetes that may have multiple causes, and inherited disorders that are caused by a single gene. Outline two different ways of using recombinant DNA technology to treat these diseases. First of all, diabetes. Alright, so diabetes, we can make insulin from GM bacteria. And single gene disorder, we can do gene therapy. Part B, a new application of recombinant DNA technology uses the genetic modification of a plant to prevent diseases. It aims to prevent the most common sources of foodborne disease, which is caused by eating food contaminated with pathogenic E. coli bacteria. Scientists genetically modifies edible spinach plant to produce colicins. Colicins are antimicrobial proteins that can kill pathogenic E. coli. Colicins are normally made by other bacteria. An experiment was carried out to test this application by spraying an extract of genetically modified GM spinach in buffer solution onto a raw meat contaminated with pathogenic E. coli. The meat was stored at 10 degrees Celsius for varying length of time before the number of viable living pathogenic E. coli bacteria were counted. Figure 4.1 shows the result. So stored at 10 degrees Celsius 1 hour. All right. We can see with no treatment, and with buffer solution only, there is almost similar log number of viable bacterial cells per gram of meat. However, buffer solution plus GM spinach decreases the bacterial number significantly. As the time passes, it benefits. Alright, buffer solution with GM spinach benefits. It decreases the viable bacterial cells per gram of meat. With reference to figure 4.1, evaluate the effectiveness of using GM spinach spray on raw meat to prevent foodborne disease. So we can give both valid and non-valid, not valid answer. So we'll, with reference to, since they asked us to give us a they told us to evaluate the effectiveness of using GM spinach. So first of all, this is valid because GM spinach does decrease the number of bacteria. As we can see, the number of bacteria is decreasing. Alright, and longer the meat is stored, the more effective the GM spinach extract is. However, it is not valid because no treatment also decreases the number of bacteria. As you can see, if there is no treatment, the number of bacteria does decrease. Alright, people are unlikely to eat meat that are left at 10 degrees Celsius for 72 hours. So the experiment does not test effect on people as well. Part 2. Regulatory authorities may not approve the use of GM spinach spray on raw meat to protect consumers. Discuss the concerns that may stop regulatory authorities approving the application of recombinant DNA technology. GM spinach spray may alter the taste and texture of meat. GM spinach spray may harm beneficial bacteria in the gut, and it may also be unsafe for humans. Example, it may cause allergies in humans. Question number 5. Photosynthesis is a complex process involving light-dependent stage and a light-independent stage. Name the products of light-dependent stage that are needed in the light-independent stage. So basically, from the light-dependent stage we require ATP, and also from the light-dependent stage we require reduced NADP. Describe the role of chlorophyll B in photosynthesis. We know that chlorophyll B can act as an accessory pigment. So chlorophyll B can absorb light energy and pass the energy to primary pigment chlorophyll A. A student carried out an experiment to investigate the effect of light intensity and light wavelength on the rate of photosynthesis. An aquatic plant, Elodia canadensis, was put into a beaker containing sodium hydrogen carbonate solution as a source of carbon dioxide. So the sodium hydrogen carbonate solution was used as a source of carbon dioxide, and we're basically doing the experiment on Elodia canadensis. To minimize changes in temperature, an LED lamp was used as a source of light. The lamp was switched on and the number of bubbles released by the aquatic plant in 1 minute was counted. The lamp was placed at 7 different distances from beaker to change light intensity. 5 replicates were carried out at each lamp distance. All other variables were controlled. So we control temperature and every other variable. The student calculated the light intensity for each distance using 1 over d square method. Alright, so using this particular formula. Table 5.1 shows the calculated light intensities for each distance. Okay. So since 1 over d square, so 1 over 0.1 whole square, so which will be equals to 100. Complete table 5.1 calculating the light intensity for distance 0.1 meter. So we already got it, it will be 100. Part C. At each distance from the lamp, the experiment was repeated using a red filter in front of the lamp to give a different wavelength of light. Okay, the experiment was repeated using a blue filter and then using a green filter. Each filter transmitted the same light intensity. The student calculated the mean rate of bubble production as a rate of measure of photosynthesis. Figure 5.1 shows the graph of the result. White light Okay, so we can see white light has the highest rate of photosynthesis. All right, then red light. All right, then we can see red light. So red light possesses a lot more energy. All right, then blue light, and then green light. Green light has the lowest because it is not absorbed by plant. With reference to figure 5.1, state the range over which light intensity is the limiting factor. Explain why light intensity above this range is not limiting the rate of photosynthesis. So we can see in every cases light intensity is limiting factor up to 400 nanometer. So from 0 to 400, from 0 to 400 light intensity is the limiting factor. After 400, concentration of carbon dioxide becomes limiting factor. A lower temperature will also limit the rate of enzyme catalyzed reaction. Low carbon dioxide concentration means less carbon dioxide will be fixed. Maximum light is being absorbed by the pigments. At a light intensity of 1600, explain why different colored filters result in different rate of photosynthesis. So different wavelength of light is absorbed by different pigments. Red light is absorbed the most. Green light is absorbed less. The more light absorbed means greater the photophosphorylation can occur. Guys, thank you for joining the video today. In our next video, we'll be uploading question number 6 to question number 10. So if you like the videos, please subscribe to our channel and hit the bell icon so that you can get notification whenever new videos are uploaded. And if you want any particular question paper to be solved, please contact us, all right, and comment in this particular video, and definitely we'll try to put that particular video as a first recommendation. Thank you. See you in the next video. Bye bye.